Hey guys, today we're going to continue on with our genetics unit, and this time we're going to be talking about non-Mendelian genetics and three particular inheritance problems: codominance, incomplete dominance, and multiple alleles. So let's get started. What are non-Mendelian genetics? Well, they are going to be the inheritance of any traits that do not follow Mendel's four principles. So if you remember, Mendel's principles said that there would be two alleles. They would be dominant and recessive alleles, that they separate independently of each other and are not linked. Well, with today's notes, and then when we learn about the second part of non-Mendelian genetics, we're going to learn about inheritance of traits that share dominance between the different alleles, that there might be traits linked to a second trait. They might rely on multiple genes or come in multiple forms. And since they don't follow Mendel's rules, there's going to be a little bit of different allele assignment and also the interpretation of the phenotypes when we look at those genotypes. So let's learn about the first non-Mendelian problem, which is a codominant inheritance. Now, when you see any problems that are codominant, that means that the two alleles that make up a genotype, the two different forms that exist, are equally dominant to each other. So instead of the dominant allele being shown or the recessive, you'll actually see both alleles expressed simultaneously. And because of that, you often see a pattern forming with the organisms. Like this cow has brown and white spots, and this chicken has black and white feathers, and this flower has red and white petals. Since you can see both traits expressed simultaneously, they are codominant. Like if you were to have co-captains of a team, they're both present on the team, they're both representing the team simultaneously. And that's what's going on here. Now, to represent the fact that this is a non-Mendelian problem, we have to assign each allele its own individual capital letter. The reason that we're using two different capital letters is because they're both dominant. So if we had a recessive allele, that would mean that that trait, that allele would be recessive and would be masked. And since there is no masking going on here, we will not have a single lowercase letter in these problems. But we do need to differentiate between the two different alleles, and that's why we write them in two different letters. So let's take the cow as an example. One allele would represent the brown coloring, and that allele would be represented with a capital B. Now that cow also has white fur or white hair, so there's also a capital W, right? Those are the two different versions, but they're both dominant, so they're both written capital letters. And so that cow has a genotype of BW. Now, anytime you see a cow or a horse that has this brown and white or red and white colored fur, that cow or that horse is known as roan. So when you see a problem that mentions a heterozygous individual being roan or having a pattern, or maybe it doesn't say roan, maybe it doesn't say a pattern, but maybe it talks about two different colors, like a black and white chicken, that is an example of a codominant problem. And this chicken is BW, not for brown and white, but for black and white. And so the flower up here is red and white. Its genotype would be RW, for red and white. Now let's try the practice. Okay, this practice says black feathers and white feathers are codominant in chickens, like we just talked about. So now we want to cross a black and white chicken with a homozygous black rooster. So since the chicken is black and white, it's heterozygous, its genotype is going to be capital B, capital W. and the homozygous black rooster, homozygous meaning the same, he's black, that genotype of the rooster would be capital B, capital B. So this is the parent cross for a black and white chicken with a homozygous black rooster. Now to solve this Punnett square, it's a monohybrid cross, so our Punnett squares are 4 boxes. We'll take the black and white rooster and we'll put its 2 alleles across the top, and we'll take the black roosters and put their alleles down the side. And then we solve the Punnett square just like we did our monohybrid crosses. But be sure to keep the allele that you wrote first in any heterozygous individuals first. Or I just like to say by rule of thumb, keep it alphabetical. Okay? Just so that you don't think there's a different genotype. Okay? So the first box would be homozygous black. The next one would be heterozygous, BW, keeping my B's first. The next box, homozygous black. And the last box would be heterozygous. So when I do my genotypic ratios, I start with the first box, homozygous black, 50% for these 2 boxes, and heterozygous, 50%. The second question asks, what are the chances they have a chick with only black feathers? So I have to interpret those genotypes. Black can only be represented by 2 dominant alleles that are both the same and black, so that would be 50%. Let's do another problem, okay? So horses can be brown, white, or brown and white, which is also known as roan. In this problem, we want to cross 2 roan horses. So since a roan horse is brown and white, it's going to be a heterozygous individual. 2 of them, BW, BW. Okay, so now let's set up the Punnett square. First parent across the top, second parent down the side, and cross your boxes. BB in the first box, BW in the second box, third box, BW, and fourth box, WW. Genotypic ratios start in box number 1. BB, 25%. Next one, BW. That's in 2 boxes, so that's 50%. And WW, 25%. When it comes to interpreting phenotypes, it goes like we did before. BB would be black, BW would either be brown and white, or you can write roan, and WW would be white. The second non-Mendelian problem of today is called incomplete dominance. Now in these problems, you're going to actually see 3 different unique phenotypes. And that's because the 2 alleles that are present in this trait are insufficiently dominant to each other. So that means that we don't just have a dominant and recessive, that's why it's non-Mendelian, and we don't have 2 alleles being equally dominant like a codominant problem. Now because they're both insufficiently dominant, they're going to blend with each other. So this is why you need to know your color blends, okay? Each allele is still represented with a different capital letter, but when you interpret the phenotypes, you're going to see a blend for a heterozygous individual. So if we look at the flowers, red is going to be dominant, so capital R, and so is white, but they're insufficiently dominant. So when you have your white flower here, genotype homozygous W, the red flower, homozygous red, and because we have a pink flower, that's actually our heterozygous individual. Same thing can apply to these mice. There are white mice, there are black mice, and then there are gray mice, BW. If you notice that these genotypes are very similar to the genotypes of a codominant problem, and that would be correct. But you need to be sure that you are interpreting the problem appropriately so that when you look at the heterozygous individual, you are not saying that they are black and white. Instead, they are gray. Okay, or they're not red and white, but they're pink. Okay, so let's do the problem. Petal color and 4 o'clock flowers are incompletely dominant. Cross a red flower and a pink flower. Now, the only way to be red is if you're homozygous, red. The only way to be pink is if you're heterozygous. So that's our parent cross, red and white. Sorry, not red and pink. Let's take the red flower and put their genotype across the top, and the pink flower and put it down the side. And now cross for our offspring. These first two boxes are both going to be homozygous red. The bottom two boxes are going to be heterozygous. So that means when I interpret my genotypes, I'm going to be 50% RR and 50% RW. So when I interpret the colors for the phenotypes, RR is red and RW is pink, 50% each.